Hello, everyone. I'm happy for the opportunity to talk today about MRI reporter genes. This is part of a webinar series on precision imaging um, of agents and theranostics. And it is sponsored by the NIH P41. My name is Asaf Gilad, and I'm going to talk about reporter genes for MRI that are designed for molecular imaging with emphasis on neuroimaging. So this slide contains reading material for those of you who are interested in reading. There are few review articles that might be useful. So the first question is, why do we need genetically encoded probes? And one reason we need those genetically encoded probes is to compensate on loss of contrast with cell division. So traditionally with MRI, people have used nanoparticles for cell imaging. And the problem with nanoparticles is that you can introduce a fixed amount of nanoparticles into a cell, but once the cell starts to divide, each of the daughter cells will inherit half of your contrast agent. So in a sense, after a very few number of cell divisions, you lose most of your contrast. Now, this is not the case if you introduce a gene into the genome of these cells. The gene will, if it's incorporated into the cell's genome, will be transferred to each of the daughter cells. So the contrast will remain if the cells divide. The other thing is that with traditional contrast agents, you cannot distinguish between live cells and dead cells, which means that even after the cell die, very often the contrast agent, especially nanoparticles, will remain in the tissue. And the last advantage of genetically encoded reporters is you can use changes in cellular function. So for example, you can put the reporter gene under a specific promoter, and you can measure cell differentiation, you can measure secretion of certain factors or response to treatment. So all of these are good reasons for us to look into use of genetically encoded reporters. So we all know that different cells express different genes. In the human body, we have different types of cells. For example, in the brain, we have various types of neurons, and also other kind of cells that are not neurons. Those could be glias or astrocytes or oligodendrocytes and so on. Now we all know that all the cells in the body contain the same DNA. But they are very different phenotypically because the DNA is translated into different proteins. So what determines which proteins will express by each cell? Well, the DNA is preceded by, or the gene is preceded by a certain fragment of DNA that is called promoter. Transcription factors are proteins that will bind to the DNA and start and will enable the binding of the RNA polymerase, and together will allow the transcription of the DNA into RNA. Later on, the RNA enters the ribosomes and it is translated into proteins. Reporter genes, as you can see here in the illustration, can be inserted into the cells using synthetic biology, and they help us to detect all these changes or to locate cells using some sort of information or some sort of readout. And this readout could be fluorescent protein, could be some sort of enzymatic reaction. The reporter genes could be transporters that help transport contrast agent or imaging probe into the cells, or they could be certain molecules that accumulate some other isotopes or ions that we can detect in the body, and so on. Now, reporter genes are genes whose product can be readily detected, and it is either fused or inserted instead of the gene of interest. The main applications of these reporter genes are: one, monitoring gene expression level; two, investigating dynamic molecular interactions between proteins; three, studying cellular interactions; four, tracking cells in normal or abnormal development; or cell transplantation therapy. Monitoring gene, and the last one, number five, is monitoring gene replacement therapy. So the revolution has started in 1972 or in the early 70s by Paul Berg and colleagues, when they performed the first molecular cloning of a gene, and essentially they took gene from one organism, they digested it with what we know today as restriction enzymes, and they paste it into a DNA of a different organism, and they were able to express a different protein in cell that naturally would not express this protein. So common reporter genes that are in use today differ depending on the imaging modality that we are using. Probably the most common reporter genes are fluorescent proteins. We detect them using fluorescence microscopy or optical imaging. Probably the most famous is the GFP, the green fluorescent protein, but there are many of them available. As another class of reporter genes are genes for bioluminescence imaging. Those genes differ from the fluorescent genes or the fluorescent proteins by that they do not require absorption or excitation of the imaging probe with light. Essentially, those are enzymes that catalyze a reaction, an enzymatic reaction that result in emission of photon. This could be detected with very sensitive cameras. And then we have reporter genes for MRI or MRS, magnetic resonance imaging, or magnetic resonance spectroscopy. And finally, we have reporter genes that are designed to be used in nuclear imaging, in PET or SPECT. And today we are going to focus on the third category, which is reporter genes for MRI. So this field has been quite active since the early 90s, with somewhat increase in activity in the 2000s. There, and this is a table from a review paper we published in 2008, and they were maybe 6 or 7 different reporter genes. In 2015, we published an updated review, and as you can see here, there are much more options for reporter genes. And the reason that we have a lot of options for reporter genes is because MRI is a very versatile tool. There are many contrast mechanisms that we can take advantage of. For example, and this was the most explored contrast mechanism, is the usage of T2 or T2 star mechanism. And in this case, we have proteins that bind or are associated with accumulation of iron. Iron is or iron oxide is a very strong T2 agent that leads to darkening of the MR image. The other options, T1 agents, those are usually enzymes that somehow involved in generation of T1 contrast or shortening of T1, mostly by some sort of manipulation of gadolinium or manganese-based agents. There are class of reporter genes that we developed in our lab that are based on CEST, on chemical exchange saturation transfer contrast mechanism. Few that were designed for spectroscopy. And finally, there are a couple of reporter genes that were suggested that are based on hyperpolarization of nuclei. So as we can see here, we have a schematic illustration, again from the same review article. And the T1 probes can work either in a manner of cleavage of a substrate and revealing of the free coordinate of the gadolinium atom, or due to polymerization of contrast agents, again, gadolinium-based contrast agents, mostly an increase or shortening the T1 of the target. T2 probes, T2 probes or T2 reporters work mostly through aggregation of iron-based compounds. This will enhance the contrast. And CES probes are more diverse. They could be either by manipulation of the reporter in a way that we have accumulation of the probe. This would lead to increase in the contrast, or could be by enzymatic cleavage of a moiety that results in reduction of the contrast. So historically, we started with MR reporter genes that were designed for MRS, magnetic resonance spectroscopy, that was done by Ellen Koretsky. And the idea is that creatine kinase is an enzyme that is expressed only in certain tissues, it catalyzed the conversion of ATP to ADP, and by that producing phosphocreatine or PCR that can be detected by MR spectroscopy. So this is, for example, when the creatine kinase is expressed in the liver, you can see the MR spectra. If you transgenically express the creatine kinase in the liver, you can see accumulation of phosphocreatine. So that was a brilliant idea. And later on, it wasn't used too much, but later on there was a follow-up study that show that you can use creatine kinase as a marker for low density lipoprotein receptor, the LDL receptor expression in LDL deficient mice after treatment with adenovirus engineered to co-express the recombinant receptor and the CDK and the CK as a reporter gene. There were several more reporter genes that came up throughout the years. Noteworthy is the arginine kinase that was developed by Glenn Walters. And arginine kinase, the concept is very similar to the creatine kinase, where the ability to phosphorylate arginine exists only in bacteria. So if you express arginine kinase in mammalian cells, you will get this additional peak of the phosphoarginine. Maybe one of the milestones that kickstart the field of MR reporter gene was developed by Angelique Louis and Tom Mead, and that was a substrate for the beta-galactosidase. So beta-galactosidase is an enzyme that encoded by the LAC-Z gene of E. coli. It catalyzes the hydrolysis of beta-galactosidase, and it is widely used in microscopy. So this enzyme has been used as a reporter gene for over 50 years, but mostly due to the colorimetric reaction that you can produce it with the right substrate, for example, XGAL. What that group did is they designed an imaging probe, and it's called eGAD-Me, where the gadolinium molecule is caged within chelator and covered with a galactose moiety. Only in the presence of beta-galactosidase, the enzyme, this galactose moiety is cleaved, and it exposes the gadolinium atom to the surrounding water, which allows exchange of water, and this exchange of water leads to reduction in the T1 relaxivity of the water, and since the exchange is very fast, it's millions of hertz, the effect is quite significant on the water contrast. As you can see here, these are cells, or not cells, this is a solution without beta-gal, but with the agent, and this is a solution with the agent and the beta-gal. So note how the beta-gal leads to brightening of the image. And they went on and demonstrated that the beta-galactosidase can be used for imaging of larvae. But that's as far as you can go with it, because the substrate supposedly has a problem that it cannot penetrate the cell wall. Another reporter system that was explored was the transferrin receptor, the ETR, and overexpression of ETR resulted in approximately 500% more of cellular uptake of transferring myon. Myon is a small iron oxide crystal and that accumulates inside the cell. And the idea is that we can overexpress transferrin and we can expose the cells or inject mice with transferrin conjugated to myon. Only the cells that expressing ETR will take up the iron oxide, and therefore we'll see reduction in the T2 contrast. As you can see here in the T2 weighted image, this is the tumor with the transferrin receptor, and it appeared to have reduction in the T2. Giving credit also to Alan Koretsky, who did some preliminary studies with the transferrin receptor quite earlier, but never published it beyond proceedings of the ISMIM. So the improved version of this kind of reporter was the ferritin, and the ferritin is the cellular storage protein of iron. That's where all the iron in the cell that is not associated with any process is stored. And it's composed of 24 sets of two proteins, the light chain and the heavy chain. They create this structure, this protein structure that can store 4,000 iron atoms per complex. And obviously, this will lead to reduction in the MR signal intensity, or in the MR shortening of the T2, a reduction in the T2 relaxation. So this is an early study by Ziv Goldfeld from the group of Michal Neiman. Early in '96, they published, and these are xenopus oocytes. So these are frog eggs that were injected with commercially isolated ferritin, and they were able to demonstrate that ferritin by itself can lead to reduction in the MRI contrast, in the T2 contrast. Later on, Michal Neiman and her group demonstrated that you can transgenically express the ferritin, and in this case, in a C6 glioma. And you can see by the image that the cells that express the ferritin has marked reduction in the T2 contrast compared to control cells. About the same time, Eric Ahrens published a similar paper where he used the ferritin as an MRI reporter gene. He used adenovirus to inject the ferritin into the brain of mouse. And he was able to show that in cells that took up the adenovirus and expressed the ferritin, there was accumulation of iron in the cells that was enough for them to be detected with MRI. 2 years later, the group of Michal Neiman came back with another paper in Nature Medicine where they had a transgenic animal that expressed the ferritin, and they were able to see the ferritin expression in a conditional manner in different organs, from embryo to the brain and the heart. This is one of my favorite MRI reporters, mostly because the level of complexity that was unusual to the MR community, and that was developed by Ben Bartel in the group of Dan Turnbull at NYU. And if we look at the construct, we have a vascular promoter that allows the reporter to be expressed only in blood vessels. Now note that beforehand, people mostly expressed it in the reporter genes in tumors, and or just injected it to the striatum and got general expression. But here they had actually cell-specific expression in the vascular system. They used a sequence, bacterial sequence that is called biotag, that is translated into a protein that is biotinylated. Inside the cells, they use the PDGF transmembrane receptor to make sure that the biotag is expressed outside of the membrane. And then they used iris allows double translation of a protein. So one protein will be the red protein and the green one will be the second protein that is translated. They are not fused, they are translated separately. And this is the BRA. BRA is the protein that biotinylate the biotag. So when the cells express this construct, the protein is produced and expressed on the cellular membrane facing the vascular, the inside of the vascular lumen in the inside of the blood vessels. When you inject avidin labeled with gadolinium, you can see that the gadolinium binds to the biotinylated biotag, and it retains on the blood vessels. So that's actually a pretty nice system, a very nice system, very elegant. And if you have time, you can read this paper. Noteworthy is another paper from Ben Bartel and Dan Turnbull about manganese-based reporter, which we don't have time to discuss. So we've been working in my lab and since the early days of my research on reporter genes that are based on chemical exchange saturation transfer, or CEST. And for those of you who are not familiar with CEST, though I think it has been discussed on one of the other webinars, the way it works is that we have our imaging probe, which is a molecule or either a protein or some sort of a small molecule that has protons that attach to the backbone of the protein. And we can saturate the magnetization of those protons. Now remember, as you remember, protons is what you detect with MRI. However, these protons rotate at a different frequency. They are rotated somewhere downfield from the water resonance frequency. So if we give a saturation pulse, a strong radio frequency pulse, we saturate the magnetization of these protons. And these protons will exchange with the water protons. So they physically jump from our imaging probe into the water, but since the exchange rate is hundreds of hertz or thousands of hertz, and we can saturate for several seconds, we have amplification of the contrast, hundreds or thousand-fold amplification based on the imaging probe, and therefore we can do the detection as a reduction in the contrast or in the reduction of the signal that we obtain from the water molecules. So to summarize, we have an imaging probe, we saturate the magnetization of specific protons with a specific frequency-dependent radio frequency or pulse, and there is a process of exchange between the imaging probe and the water molecules, and we can detect the transfer of the magnetization in the water signal intensity. The way we generate the image, we give the saturation at a specific frequency, let's call it delta or small delta, and we get reduction in the MR water signal intensity. Then we saturate the compound at the opposite side with a frequency that is shifted from the water frequency, but in the opposite side, so upfield of the water frequency. This should not result in any reduction in the water frequency, or maybe very small due to NOE effect. And we subtract them, and what you can see from the subtraction is the contrast that is generated by the CEST probe. So the first experiment that we did was to design a reporter gene based on CEST. We knew that polylysin is a good CEST agent, and therefore we designed the LRP, the lysin-rich protein. We generated two tumors. One expresses EGFP, or green fluorescent protein. One expresses the LRP, and transplanted them in a mouse brain. When we applied a CEST pulse sequence, we can see that we can detect there are cells that are expressing the LRP. From that on, we kept working with the LRP with different promoters. We share it with other labs. So for example, the group of Moriel Vandesberger showed that they can use it to track gene therapy in the myocardium and the heart muscle. We showed that we can use the LRP to detect specific promoters in tumor. And Chris Farrar from MGH was able to use it to track oncolytic virotherapy. So the last example that we're going to elaborate on is using nucleoside kinase as MR reporter gene, and some of you are familiar with this kind of reporter genes from the HSVTK that is quite frequently used for PET. So in all cases, the HSVTK is an enzyme, the herpes simplex thymidine kinase is an enzyme, a viral enzyme, that can phosphorylate thymidine. Thymidine is one of the building blocks of the set of the DNA. That's the T in the ACTG. And what's interesting is that thymidine can go inside and outside the cell freely, it diffuses across the membrane. But when it is phosphorylated by thymidine kinase, either the endogenous thymidine kinase or the viral thymidine kinase, it is trapped inside the cell. So phosphothymidine is trapped inside the cell. And free thymidine or unphosphorylated thymidine is free to go. What's unique about the HSV-TK is that it can phosphorylate thymidine analogs that cannot be phosphorylated by endogenous thymidine. For example, acyclovir, ganciclovir, those are antiviral drugs, but also FIAU, which can be iodinated, and it's a reporter gene for PET or SPECT. So Amnon Bashir, who is a professor at the Weizmann Institute, back when he was in my lab, designed a probe that produces cis contrast when it is trapped inside cells, and he engineered tumor cells to express the thymidine kinase, the HSV-TK. Upon injection of the cis probe, we can see that the probe accumulates both in the HSV-TK expressing tumor, but also in the wild type tumor and the control tumor. And this is due to extravasation of the probe from the leaky blood vessels in the tumor. But with time, it is washed out of the tumor and it stays only in the cells where the HSV-TK phosphorylates the imaging probe. And we validated it with the help of Sridhar Nimigada. We validated it in with SPECT-CT in histology. So finally, we used a variant of a thymidine kinase. This is actually a deoxyribonuclease kinase. This is a similar enzyme from the Drosophila, the fruit fly, which is even more permissive than the HSV thymidine kinase in a way that it phosphorylates a larger variety of imaging probes. So again, the same principle, the imaging probe, in this case, it's called Pyrrolo-DC, can enter inside and outside the cells, but only in the presence of the DMDNK, the enzyme. It is phosphorylated and trapped inside the cell. This is the imaging probe. This is the exchangeable proton, phosphorylated and unphosphorylated form. What's special about it is those rings allow optical detection. As you can see here, we can detect the cells that take up the thymidine, they take up the imaging probe. So we designed a sophisticated experiment where we labeled dendritic cells. We engineered them to express the DMDNK as a reporter gene. We started by injection of the engineered dendritic cells into the footpad of a mouse, together with stimulation that caused some sort of allergy, and the dendritic cells migrated to the popliteal lymph node. After several days, we injected the imaging probe, the Pyrrolo-DC, and we were able to see accumulation of those cells in the lymph node, which we could not observe in cells that did not express the DMDNK. So what's interesting about it is that we can quantify it because, as we said, the Pyrrolo-DC, the imaging probe, is fluorescent, and we can do cell sorting. We can take out the lymph node, we can dissect it, and we can count the cells. and we were able to calculate the absolute number of cells, which was on average 8,155 cells per lymph node. So I'm not going to discuss due to the shortage of time, but there are a few nice studies from the group of Mikhail Shapiro from Caltech, where they used hypercest to detect genetically encoded gas vesicles. And a recent paper in Science, they were able to express a whole set of genes in mammalian cells and can detect them, not sure if with MRI, but definitely with ultrasound. So in summary, the motivation of developing reporter genes for molecular imaging is to assist with temporal studies that employ molecular biological assays that usually requires large number of animals. And if you use reporter gene, you can track the same animal longitudinally, so over time, and in a non-invasive manner. And reporter genes can be used in engineered cells, transgenic animals, and in cell and gene therapy, and for cell and gene therapy. So with that, I would like to thank you for your attention.